So with that in mind, I want to introduce tonight's speaker, who is from the J. Craig Venter Institute here in San Diego, which I suspect many of you already know, made a recent report that they had made a major breakthrough in synthetic genomics. And our speaker, Bob Friedman, is the California director for the Venter Institute. And what he's going to do is tell you something about what that means, what the Venter Institute is, what synthetic genomics is about, and then that will lead into a conversation about some of the ethical challenges that go with that. So on that note, Bob, it's your program.
Thanks very much. And let me see if I can actually get us to my presentation. It's always a little bit of a challenge. There we go. Okay. As Mike said, I'd like to talk to you this evening, and I appreciate everybody's attendance here on synthetic genomics, risks and benefits for science and society. And I'd like to cover in a perhaps whirlwind tour a few topics this evening. First, a few words about the Venter Institute, who we are. I will give a brief introduction to the technology, and probably more interesting than the technology, its potential applications, an overview of the societal issues that it raises, and then I will get to the subtitle that you saw before, Who is Watching, talking a little bit about the current regulatory system and some new initiatives in the United States and Europe. So just starting with the Venter Institute, we're a nonprofit research organization. We're about 350 scientists and staff in Rockville, Maryland, and here in San Diego. We do all things genomics. Genomics is just what can you learn by studying the DNA of an organism. Turns out you can learn a lot. So we work in areas of genomic medicine, infectious disease, applications of genomics for understanding the environment, and synthetic biology, which I'll talk about a little bit today. We also have an education mission, and we undertake policy research, and I direct the Policy Center at the Venter Institute. Our headquarters in Rockville, Maryland. In San Diego, we're off Science Center Drive, and I can't resist but to show a building on the drawing boards, which we hope to build on the UCSD campus, which we hope will be the first zero carbon laboratory facility in the world. So, what is the glue that binds all the research at the Venter Institute together? It is DNA, the study of DNA. And here on the bottom is a picture of a DNA molecule. These are the bases, the A, C, G's, and T's, the letters of genetic code. And much of the activity within our institute is sequencing DNA, trying to decode the chemical composition into, again, these series of letters of bases, A, C, G's, and T's. We can also go from this digital information and synthesize, resynthesize this this chemical, this piece of DNA that you see on the bottom. So sequencing one way, synthesis the other. Just to give you an idea of scale, these bases, these nucleotides, a single gene is perhaps hundreds to thousands of base pairs. A gene is a unit of DNA that can perform a useful function within a cell. The genome, that is the complete DNA of an organism. For a virus, it's quite small. It's thousands to hundreds of thousands of bases. The bacteria that Mike referred to, the synthetic cell that Mike referred to earlier, is about 1 million bases long. The average bacteria is about 5 million bases. Human, 1,000 times larger. 6 billion. Plants can be even larger than that. The importance of scale is The technology is at the stage that we can build viruses and bacteria. We are way far away from humans, plants, other animals. So keep in mind in my comments today, we're talking about primarily bacteria, but there are lots of things that you can do and learn from bacteria. So, definition, what is synthetic biology or what is synthetic genomics? So sort of the techie baseline would be, it's really the construction of long strands of DNA, gene length or genome length pieces of DNA using chemicals. We use the term synthetic biology when we're talking about synthesizing perhaps pieces multiple genes long. Today's genetic engineering, recombinant DNA technology, typically works with a gene, maybe 2 genes. Synthetic biology, up to a dozen or 15. Synthetic genomics is working on the entire genome, again, virus or bacteria size, including activation of that genome into a cell. So simply, synthetic biology or synthetic genomics are more powerful and more precise forms of genetic engineering. Why are we interested in this new technology? Why did we pursue the development of this new technology of synthetic genomics? Well, the answer is, I think, twofold. One, this ability to synthesize DNA from the genetic code in a computer, build what you'd like, makes genetic manipulations that are impossible to do today within our grasp. In fact, we can now synthesize genes, whole chromosomes, cheaply and rapidly. Again, 2 reasons we would like to do this. One is the tremendous potential for new health and industrial applications, and I'll go over a few of those. And the second reason, and they are quite related, is that this new technology can help us understand basic biology. We will not achieve these new health and industrial applications unless our biology, our understanding of biology greatly improves. So our ability to manipulate DNA today far exceeds our ability to understand how cells work. Synthetic genomics will help us in that task of understanding how it works. So a few applications, one that you may have heard of, a small molecule therapeutic called Artemisinin. As I think most of you know, malaria is the leading cause of illness and death worldwide. There's a company up in the Bay Area that is now synthesizing artemisinin, or a precursor to artemisinin, a very potent anti-malarial drug that is now today harvested from a woody shrub. It is one of the least expensive alternatives available, but still too expensive for those who need it most. This company with Gates Foundation funding has taken this artemisinin synthesis pathway from the plant, put it in yeast, and has turned yeast into a mini pharmaceutical manufacturing plant. Second example, and there are many, many examples all over San Diego, lots of efforts focused on trying to develop substitutes for petroleum. The idea is to use bacteria, perhaps algae, to produce transportation fuels, carbon neutral fuels that can substitute for petroleum. This is just a cartoon from one of these companies' websites, LS9. It's a fermenter filled with their proprietary microbe. They can put in renewable feedstocks, cellulosic materials, plant stuff that grows out in the sun. The bacteria chews up the plant stuff and releases oil which floats to the surface, and that can be made into a transportation fuel. Again, Mike mentioned that we had recent, very significant achievement of being able to, and we can talk further, I will talk further about what this means, synthesize a synthetic cell. In fact, what we did was we synthesized a chromosome, the operating system of a bacteria. We are then able to coat this molecule of DNA, have the chromosome of DNA, the molecule of DNA, enter an existing bacterium, take over the function of the cell, and basically transform the cell from its original species to the new species that is controlled by this piece of DNA, this bacteria. Now, I should add, we made very few modifications. This was a proof of concept, a proof of principle that this could be done. And I know some of that sounds a little incredible, so I will go over it again. Basically, what we do is we start out with a natural cell. We take out the DNA. We put it in our sequencers. We get the DNA sequence, that digital code that goes into national databases. We can take that genetic sequence, perhaps make a few modifications, and we did make a few modifications to the original code, synthesize the chromosome, use yeast to actually assemble all these little pieces, and then we're back to what I just showed in the last slide. We have a synthetic genome, we transplant it into a cell, and the new cell, which we call the synthetic cell, takes on the characteristics of the synthetic genome. Now, though synthetic biology, synthetic genomics, synthesizing DNA has achieved some, one of the slides that was shown before was how quickly public recognition is moving of this new field of synthetic biology or synthetic genomics. It actually started our ability to synthesize DNA back in the late 70s. Scientists were able to synthesize a piece of DNA, perhaps a couple of hundred bases long. By the early 90s, we were able to synthesize a gene. By about 2000, we were able to synthesize a virus-size piece of DNA. And today, we're able to synthesize this 1 million base pair chromosome of small bacteria. Now, I'm sure many of you recognize that external events are very important as to how a technology is perceived. And one of those external events that I think really changed the way at least policymakers in Washington looked at this new technology was 9/11/2001. In fact, the first major milestone after 9/11 was the chemical synthesis of poliovirus by Eckhard Wimmer and his colleagues at the University of New York at Stony Brook. And they did want to make a point. They wanted to make a point that if scientists in the lab could synthesize a pathogenic virus like poliovirus, so could a bioterrorist. And when this paper was published in Science, again, about 11 months after 9/11, you better believe that the policy community in Washington started paying attention. And that, I think, is a good lead-in to the next part of my talk, which is really a focus on what are the societal issues raised by this new technology. I think it's fair to say that along with the promise of benefits, all new technologies raise societal concerns, and synthetic biology, synthetic genomics is clearly no exception to this rule. There are 5 key societal concerns that we at the Venner Institute have been focusing on: bioterrorism, worker safety, our folks in the lab, potential harm to the environment, human health concerns, and some ethical and religious issues. And I'll go through each of those and try to identify first what are the societal issues, and then what is different about synthetic genomics than, call it plain vanilla genetic engineering that we've been doing since 1975, products of genetic engineering certainly since 1985. So, bioterrorism. I talked about the societal issue that this new technology raises. This is a new way for a potential bioterrorist to obtain a pathogen. No longer can we just limit access to harmful pathogens by physical means, keep them locked up in a laboratory, but we now have to look at other ways to obtain the DNA that can be used to make those pathogens, viruses in particular, this new technology of synthetic genomics. Now, most pathogens are easier to obtain by other means. Steal them from a lab, get them in nature. There are a whole host of ways that a bioterrorist might access these new technologies, and Homeland Security and others are well aware, and there are many procedures in place to protect the general public. But there are a few viruses that this new technology might actually be easier. 1918 flu, smallpox, and Ebola. And clearly, those are pathogens of concern. Second issue, worker safety. I should just start out by saying the synthetic DNA itself is not harmful. That's not the concern. The concern really is that what's the risk to workers from the specific viruses or bacteria that they're working with? What's different about synthetic genomics? Well, synthetic genomics, synthetic biology are bringing a whole new group of researchers to this field. That comic book that you saw on the slides before, this was produced within a bioengineering department. We're now seeing undergraduate engineers turning to biology, perhaps treating biology in the same way as if it were a pile of integrated circuits, resistors, capacitors, and so on. They haven't gone through the more traditional microbiology training where they learn a healthy regard for biology. So some cultural changes in education in other places. Concerns for harm to the environment. Again, no concern for harm from the synthetic DNA itself. The concern is whether a specific engineered microbe might pose a risk to the environment. And this is truly part of a larger debate that's been going on about recombinant DNA technology, genetic engineering, since the mid-70s, starting with a famous conference at Asilomar. What's different about synthetic genomics? Well, again, speed and scale. We can now build new products not possible with the current genetic engineering technologies. Human health, I might just say ditto. It's not concern for harm from synthetic DNA itself. It's will we see new pharmaceuticals, cosmetics, food products, so on and so forth. And can we adequately test for the safety of those new products? Ethical and religious issues. Well, honestly, some view this research as, and this quote was on the cover of Newsweek, quote, playing God, part of a larger concern about the extent to which humans should be modifying life, and to some even studying, understand the basic principles of life. Another concern that I've heard expressed, concerns about once one uses organisms as can experiment with organisms at will, does this change the relationship of humans to nature? Is there anything new here about synthetic genomics? Well, I guess I have to say yes, I really think there is, that the construction of a free-living organism from chemicals, that synthesizing that chromosome and have it take over the function of a cell is different, is qualitatively different. And this was the cover of The Economist magazine after our Science magazine paper was released in May, and Man-Made Life, the first artificial organism and its consequences. That brings me to the who's watching part of the talk, governance. I guess in a phrase, lots of people are watching. We have a regulatory framework that was established in 1996 when the first products from biotechnology were first appearing. I'm sorry, that should say 1986 when the first products of biotechnology were appearing. You know how many times I looked at that slide? And I apologize as well for the alphabet soup of laws and regulations, and I'll just explain what that means. If the product is a microbe, the Environmental Protection Agency regulates it under TSCA, the Toxic Substances Control Act. If it's a food, Food and Drug Administration, FDA regulates it. If it's a plant, USDA regulates it, so on and so forth. And the National Institute of Health, with these recombinant DNA guidelines, is responsible for setting guidelines for safe use in the laboratory. The real question here is not who's watching, but how well are they watching? Do the laws that we established, some of these even prior to 1986, and which we reprogrammed to also cover the products of genetic engineering, are they applicable to those new products of synthetic biology? And at my center, we have, fortunately, a new study funding from the Department of Energy, a 2-year study to actually look in great detail at each of 11 pieces of legislation that are relevant to synthetic biology and try and answer that question in some detail. Look for the cracks, look for the flaws, look for the holes where our current regulatory structure may not work well. There's also been a series of recent governance efforts really focusing on this issue of how do we prevent this technology from being used by a potential bioterrorist. And there are lots of folks who have engaged in this discussion. The synthetic biology community itself at a series of almost annual meetings that they hold, several National Academy of Science studies in 2006. The federal government set up a National Science Advisory Board for Biosecurity with one of its working groups focusing specifically on this new technology. The industry that manufactures synthetic DNA has proposed self-regulation standards, and there are now actually a couple of Federal Register notices pending of new guidance on how to deal with this new technology. So a lot of attention is happening right now. We're right sort of in the middle of this change. Several years ago, we were fortunate to receive funding from the Alfred P. Sloan Foundation, along with our collaborators, the Synthetic Biology Group at MIT, and the Center for Strategic and International Studies, International Studies Group think tank in Washington, D.C., to look at this issue of options for governance for synthetic biology. And we developed a series of options to present to decision makers in Congress, the administration, industry decision makers to do 3 things. We hope: enhance biosecurity, protect the environment, local communities, and to foster laboratory safety. And we focused on a series of places that it's important to perhaps set guidance or regulations. And one of those, and probably the most germane, the most important, is is looking at what the commercial firms that synthesize DNA, produce this stuff for scientists all over the world, businesses, what can they do to keep it in the hands of legitimate users? And having worked in Washington for a long time, we sort of put together this consumer reports scheme of how do you evaluate each of these options. Can you screen the genetic sequence? Can you screen the people who are doing orders, so on and so forth? Does it enhance biosecurity and so on? And as a result of this work, and I'd be happy to go into great detail on these, but I think the bottom line is that as a result of our work and the work of others, because there are many others who were working along with us, there is, as I mentioned now, guidance proposed by the Department of Health and Human Services asking these companies that synthesize DNA, again, sell to legitimate workers, to do two things: to screen these chemicals to make sure that these orders aren't being placed to synthesize a pathogen, and to screen who's, unless you are a legitimate scientist studying these pathogens, and to screen the users, the people who are placing the orders. So I think after several years of effort, there is a good end to this story. And this is not a Rorschach test, but just our complete scheme of the various options and that we propose, focusing on people who synthesize big pieces of DNA, small pieces of DNA, the machines that do this, and the users themselves. And if anyone is interested, I'd be happy to mail you a hard copy, or it is available online at the Venner Institute website, jcvi.org. I think the report is still current, and we'll give you sort of an in-depth look at the type of policy work that has been focused on this new technology. And with that, I'll just close with this slide, just put up again what we think are those 5 key societal concerns: bioterrorism, worker safety, harm to the environment, human health, and ethical and religious issues. And thank you for your attention, and be happy to answer any questions.